Hello everyone and welcome to my first A-level biology video. So this video is going to be on protein synthesis for AQA AS and A-level biology. So the first specification points include number 1, nucleic acids carry the genetic code for the production of proteins. Amino acids are the monomers from which proteins are made. 2, the base sequence of each gene carries the coded genetic information that determines the sequence of amino acids during protein synthesis. So for our first specification point, here we have a nucleotide, and nucleotides are monomers that make up nucleic acids, which are polymers. Now, a nucleotide is made up of a phosphate, a pentose sugar, and a nitrogenous base. So the pentose sugar could, for example, be deoxyribose, which is the pentose sugar in DNA, or it could be ribose, which is in RNA. And the nitrogenous base include A, C, G and T for DNA, whereas it's A, C, G and U for RNA. So here we have the structures of RNA and DNA, which are both nucleic acids, and they are polymers of nucleotides. So DNA is obviously made up of 2 strands of nucleotides bonded together, whereas RNA is 1 strand. So amino acids, which are monomers, actually make up new proteins, which are polymers during protein synthesis. So for our second specification point, here we have our DNA molecule with our gene selected, and genes made up of bases, so A, T, G and C. And it is in fact the triplet code which determines the order of amino acids. So 3 base sequence codes for a particular amino acid. So for example, we've got TAC here, which codes for this particular amino acid, CGA, which codes for this particular amino acid, TGC, which codes for this one, and so on. So it is this base order which controls the order of amino acid assembly. So the amino acid sequence also determines the protein produced. So now some questions based on that information. So question 1, what is a monomer? So we know that a monomer is in fact a small molecule which makes up large molecules or polymers. So here we have our answer. So a monomer is a smaller or repeating unit or molecule from which larger molecules or polymers are made. Question 2. Table 1 shows mRNA codons and the amino acids coded for by each codon. It also shows some properties of the R group of each amino acid. And then the question is, the genetic code is described as degenerate. What is meant by this? Use an example from table 1 to illustrate your answer. So although I haven't specifically gone over what degenerate actually means, we have discussed the specific answer to this. So the genetic code being described as degenerate means that more than one codon codes for a single amino acid. So for example, if we have a look at the table, so we've got, for example, pro here, so proline. So if we have a look at our first base, so C, C and U codes for proline, and also C, C, C and C, C, A and C, C, G. So those 4 codons all code for proline. So we could use that as our example, for example. So for our third specification point, we need to know the structure of molecules of messenger RNA and of transfer RNA. We need to know that transcription as the production of mRNA from DNA, the role of RNA polymerase in joining mRNA nucleotides. In prokaryotes, transcription results directly in the production of mRNA from DNA. In eukaryotes, transcription results in the production of pre-mRNA. This is then spliced to form mRNA. Translation as the production of polypeptides from the sequence of codons carried by mRNA. The roles of ribosomes, tRNA and ATP. Students should be able to relate the base sequence of nucleic acids to the amino acid sequence of polypeptides when provided with suitable data about the genetic code. Interpret data from experimental work investigating the role of nucleic acids. Students will not be required to recall in written papers specific codons and the amino acids for which they code. So first we're going to be discussing transcription. So here we have our DNA molecule, and an enzyme which is known as DNA helicase breaks the hydrogen bond between the DNA strands and the DNA molecule unwinds. Now, with all the A-level specifications, they don't mention you needing to know about DNA helicase for protein synthesis. However, they do mention it when talking about DNA replication. So I'll just check with your teacher as to whether they think that you need to include DNA helicase in your protein synthesis answers, or whether you just need to say that the hydrogen bonds between DNA break and the DNA molecule unwinds. And then after this, the DNA strands separate, and there are two DNA strands. So there are the coding DNA strand, and there is the non-coding DNA strand, also known as the template strand. So just to remember as well that transcription occurs in the nucleus in eukaryotes, but of course prokaryotes do not have a nucleus, so transcription occurs in the cytoplasm. So the non-coding / the template DNA strand is actually the strand used for the copying of the code to mRNA. So the template strand is almost a reflection of the code from the coding strand, and then mRNA copies this code from this strand. Next, an enzyme called RNA polymerase lines up 3 complementary RNA nucleotides alongside DNA's template strand. Hydrogen bonds then form between the bases. So just a reminder that bases of DNA are A, T, C and G, and RNA bases are U, A, G and C. A complementary base pairs with U with 2 hydrogen bonds. T complementary base pairs with A with 2 hydrogen bonds as well. Then we've got C complementary base pairing with G, and G complementary pairing with C, and they are 3 hydrogen bonds. So once all the RNA nucleotides are in place, hydrogen bonds between the bases break, and RNA polymerase joins the mRNA nucleotides, forming the mRNA strand. So for prokaryotes, the mRNA strand is formed immediately, but for eukaryotes, a pre-mRNA strand is formed. So in prokaryotes, transcription and translation are actually coupled. So hydrogen bonds break between the mRNA and DNA bases, and the mRNA immediately associates with ribosomes and translation begins. Because there's no nucleus to separate transcription and translation, the prokaryotic genes are transcribed and transcripts are immediately translated. So in eukaryotes, there are actually post-transcriptional modifications which occur to the pre-mRNA strand produced. So transcription results in the formation of the pre-mRNA, and pre-mRNA is made up of exons and introns. And exons are coding regions, whereas introns are non-coding regions. So since introns are non-coding, they are removed and exons are spliced together in a post-transcriptional process known as splicing. And then you get the formation of your final mRNA strand. So now we're going to discuss the structure of messenger RNA. So messenger RNA, it has a sugar phosphate backbone which forms the nucleic acid structure composed of nucleotides. The backbone is composed of alternating sugar and phosphate groups, and this defines the directionality of the molecule. So as we know, mRNA also has codons, which are triplets of bases, so sequences of 3 bases that code for specific amino acids. Hydrogen bonds form between these DNA bases and mRNA bases during transcription. So now we're going to discuss translation, and this occurs in the cytoplasm. So when mRNA leaves the nucleus, it then attaches to the ribosome in cytoplasm. Now, ribosomes play an important role during translation. They are the site of protein synthesis, and they are also the place where mRNA attaches, whilst tRNA brings amino acids to form the polypeptide. So tRNA has an important structure and an important role. So it is a folded structure with three hairpin loops. So it almost looks like a clover leaf structure. It also has an amino acid binding site at the top of one of these loops, and it has an anticodon at the bottom. It also contains hydrogen bonds between complementary base pairs within the structure, and it is made up of nucleotides. So its role in translation is binding to mRNA codons with its anticodons. So it brings specific amino acids to the mRNA in the ribosome coded by the mRNA codons. So once the mRNA has attached to the ribosome, a tRNA molecule brings an amino acid to the mRNA. So it complementary base pairs to the start codon AUG on the mRNA strand with its anticodon UAC. Then a second tRNA molecule carrying a corresponding amino acid complementary base pairs to the second codon on mRNA with its anticodon. So an enzyme then catalyzes the formation of a peptide bond between these amino acids using ATP. And the enzyme continues to do this as more tRNA molecules bring amino acids to the mRNA. So this process continues until tRNA reaches a stop codon and the polypeptide chain of amino acids detaches from the ribosome. So now we're going to move on to some questions based on that information. So question 1, give 2 structural differences between a molecule of messenger RNA and a molecule of transfer RNA. So we've discussed the structures of both these molecules and we know that there's a lot of differences between them. So we can start by saying that mRNA is linear, it's a straight chain. So that means that it doesn't have hydrogen bonds. Whereas tRNA is obviously a clover leaf structure and it does have hydrogen bonds and base pairing. We could also say that mRNA doesn't have an amino acid binding site, where you know, of course, tRNA does. mRNA as well actually has more nucleotides. It's a larger structure than tRNA. And there are different mRNA lengths as well, whereas all tRNAs are very similar or the same length. And we also know, of course, that mRNA has codons, whereas tRNA has an anticodon. So that's a lot of points I've given you there. So if it was me, I think I'd go for the 2 simplest ones, really. That mRNA doesn't have an amino acid binding site, but tRNA does, and mRNA has codons, whereas tRNA has an anticodon. I feel those 2 are the easiest to remember, really. So we've got our answer there. Question 2, starting with mRNA in the cytoplasm, describe how translation leads to the production of a polypeptide. Do not include descriptions of transcription and splicing in your answer. So this question is basically just asking us to describe translation. So we know that the first stage is the ribosome attaching to the mRNA, and then the ribosome finds the start codon AUG, and then tRNA brings the specific amino acid to the mRNA on the ribosome, and tRNA's anticodon complementary base pairs with the codon on mRNA, and then the ribosome moves along to the next codon. So then this process is repeated and the amino acids are joined by peptide bonds, and this is using energy from ATP. So here we have our answer. So there's a lot of different phrases you could put. This is a really good question practice. Question 3. CENPW is involved in the formation of spindle fibres in mitosis. Spindle fibres are made of molecules of protein called tubulin. The scientist treated cells in a culture with small interfering RNA, siRNA. This siRNA causes RNA interference of expression of the CENPW gene. The scientists took samples of cells at 0, 48 and 72 hours after adding the siRNA. They then used gel electrophoresis to separate CENPW and tubulin from these samples. Figure 5 shows the results of the electrophoresis. The size of each band is proportional to the amount of CENPW or tubulin present. So then we've got a diagram there. And then the question is, suggest how siRNA produce these results. So we can see from the diagram that tubulin production decreases. So the size of the band decreases, as does CENPW. So we can say that the siRNA must bind to mRNA or destroy it for CEMPW. So then this, of course, is preventing translation of CEMPW, and then the CEMPW will reduce. So tubulin production will also reduce. And we can see our answer there. So question 4, the oxidation of ammonia by nitrifying bacteria involves the enzyme ammonia monooxygenase. Each species of nitrifying bacteria has its own specific AMO-A gene that codes for production of ammonia monooxygenase. In a second investigation, the scientists determined the expression of the AMO-A gene in 2 species of bacteria, S and T. Species S was from acid soil and species T was from soil with a neutral pH. The scientists grew cultures of each species separately in soil with a different pH. They determined the amount of mRNA from the AMO-A gene in each culture. Their results are shown in figure 7. So figure 7 is a graph and it shows species S from acid soil and it shows the copies of mRNA per gram of soil at different pHs, and it also shows species T from soil with neutral pH and the copies of mRNA per gram of soil at different pHs of soil. So the question is, this method allowed the scientists to estimate the expression of the AMO-A gene in each culture, but not the growth of the bacterial population in each culture. Explain why. So this question obviously includes a lot of information. It seems very, very confusing. So the first thing we're going to do is definitely go through and highlight the key words. So the oxidation of ammonia, blah, blah, blah. So we've got a gene. So this gene is coding for the production of a specific molecule. And the scientists are determining the expression of this gene in 2 species. And species S is in acid soil, so S, acid soil, and species T is in soil with a neutral pH. And then the scientists are growing cultures of each species separately in soil with a different pH, and they're determining the amount of mRNA from the gene. So we know this is to do with protein synthesis because, of course, mRNA is produced during transcription and it's a copy of the gene. So we need to determine why the scientists were able to estimate the expression of the gene but not the growth of the bacteria population. So if we have a little look at the graph to help us as well, so we know in the information that they didn't actually count the bacterial cells in the species beforehand, so therefore they're not going to know how many cells have, how the cell numbers have increased in number. So that could be our first point. So that's fairly simple. They haven't counted the number of bacterial cells, so they don't know the population. And then we know that they can estimate the expression of the gene because we can see the number of copies of mRNA. So if we have a look at our graph here, we've got the copies of mRNA for species S and species T. So we can see the number of mRNA strands related to the amount of AMO-A produced or translated. So we can also say that we don't know how much mRNA is produced per cell. So we know how much is produced for the species itself, but we don't know how much is produced for the specific cell, so therefore we don't know the growth of the actual bacterial population. And then finally, we can also say that we don't know if AMO-A mRNA is actually linked to the cell division or the growth of the population, because the production of this specific molecule may be to help the cell do something different. It may not be for growth of the cell. So we have our answer there. So I understand it's a tricky question. So if you want to go back, pause the video, slow me down a bit, read it through a few times to try and get an understanding of that. And then hopefully this will be helpful for you in the exams. Question 5. So multiple copies of the AMY1 gene is an adaptation to a high starch diet. Use your knowledge of protein synthesis and enzyme action to explain the advantage of this adaptation. So we know that if there is more copies of the gene, then therefore that means that there's going to be more copies of mRNA produced. So there's going to be more mRNA molecules produced, which are copies of this gene, and therefore there's going to be more mRNA translated, and so therefore there's going to be more starch produced quicker. So there we can see our answer. So there's more mRNA or more transcription, so there's going to be more translation or more enzyme, and so the reaction is going to be faster. Question 6. Describe how mRNA is produced in a plant cell. So some of the keywords here are, of course, plant cell. So we know that that's a eukaryotic cell. So we must have to include the fact that splicing occurs after the pre-mRNA is formed in order for the final mRNA molecule to be produced. And the fact that we need to describe how mRNA is produced is telling us that we need to describe transcription to begin with as well. So we know the first stage of transcription is the DNA strands separating, and this is due to the hydrogen bonds breaking between the complementary DNA bases. And then the template strand is the one that is used to make the mRNA. So complementary base pairing occurs between the template strand bases, and the RNA nucleotides. So A pairs with U, T with A, C with G, and G with C. We can then also say that RNA polymerase joins the RNA nucleotides together in order to form pre-mRNA. And then we know that splicing occurs, which removes the introns in order to form mRNA. And here we have our answer. Question 7. Describe the role of a ribosome in the production of a polypeptide. Do not include transcription in your answer. So we know that, of course, ribosomes are involved in translation. So we need to be discussing translation here. So at the beginning of translation, mRNA obviously binds or attaches to the ribosome, and then the ribosome actually allows for 2 codons of the mRNA to fit inside of it. Then the tRNA with anticodons comes along and it binds to the codons on mRNA, and then the ribosome catalyzes the formation of peptide bonds between the amino acids, which are held by the tRNA molecules within the ribosome. And then the ribosome moves along to the next codon on mRNA, and the process repeats. So we can see our answer here. Question 8. Table 1 shows the base sequence of part of a pre-mRNA molecule from a eukaryotic cell. Complete the table with the base sequence of the DNA strand from which this pre-mRNA was transcribed. Now, I think this is a really nice question because if you can remember which bases pair with which, then this is a really easy mark to get. So we've got A to begin with, so we know that within DNA there is T, C, A and G bases, and we know that T is the base that complementary base pairs to A in mRNA. So the first one is going to be T, so T will go here. Then we've got C, so C is going to complementary base pair with G. Then G with C, C with G, A with T, U with A, U with A with T, and U with A. So we can easily do that one. So there's our answer there. Question 9. Artificial fertilizer is used to replace mineral ions removed from the land when crops are harvested. One of the mineral ions is nitrate. Give 2 examples of biological molecules containing nitrogen that would be removed when a crop is harvested. So we know a lot of biological molecules that contain nitrogen at this point. So of course we know that nitrogen is a key component of amino acids, so we can say amino acids. We also know that nitrogen is a key component of nucleotides in the nitrogenous base, which is so nucleic acids, DNA or RNA. So those are some examples, and there's also other ones put here that we haven't discussed in this particular topic, but that could also be used. Question 10. Contrast the structures of DNA and mRNA molecules to give 3 differences. Now, I know that this particular presentation isn't specifically based on DNA and RNA structures. However, we have discussed the structure of mRNA, and consequently, through discussing protein synthesis, we have touched on the structure of DNA, so we still are able to answer this question. So what we can say, based on what we've learned, is that in DNA, it has a thymine base, a T base, whereas within RNA, it has a U base or a uracil base. We can also say that DNA has base pairing due to it having 2 strands, whereas mRNA doesn't. And we could also say that DNA has hydrogen bonding and mRNA doesn't. And we also know that DNA has introns, or non-coding sequences, which our final mRNA molecule doesn't have. We also know that DNA is double-stranded, whereas mRNA is single-stranded. And we haven't touched on this, but DNA is actually very long and RNA is shorter. And DNA is made up of deoxyribose, whereas RNA has a ribose sugar in it. So those are just some examples there. So you only need to choose 3 for this specific question, as it's worth 3 marks. So that's our answer there. Question 11. Messenger RNA is used during translation to form polypeptides. Describe how mRNA is produced in the nucleus of a cell. So this is from a specimen paper actually, and it does include the fact that helicase breaks the hydrogen bonds. So this is why earlier I mentioned checking with your teacher whether you need to know about helicase or not. But for this question, we'll just pretend that we do. So we know that helicase actually breaks the hydrogen bonds between the DNA strands, and the DNA strand used is the template strand. And the RNA nucleotides are attracted to these exposed bases on the template, and this is according to the base pairing rule. So we know A pairs with U, C with G, G with C, and T with A. So then we can say that the RNA polymerase enzyme is the one that joins these RNA molecules, RNA nucleotides together, and then the pre-mRNA is spliced to remove introns. So I feel that this question is sort of similar to the other question we did. The other question was included in some actual papers, so I would probably say to base your answers based on that other question we did, but this is just another question which is good practice basically. So we can see our answer there. So thank you so much for watching. Please do go and subscribe to my channel and make sure to click the little bell so you'll get notifications of when I've posted. Also, please give this video a big thumbs up and share with any friends or people that you know that this sort of content would really help, and make sure to comment down below any more videos you would like to see, whether they are to do with biology or whether they're just general study techniques or things like that. Also, please go over to my Instagram and give me a follow, and visit my website in order to access loads more exam questions and revision resources for AQA, OCR and Edexcel, GCSE and A-level biology and combined science courses. So thank you again everyone, and I'll see you in my next video. Bye guys.